Welcome to MG Sahu Education Blog. Welcome to Sahu. In our last lesson, we began exploring the invisible world of microorganisms. We saw how tiny bacteria, fungi, and protozoa exist all around us, completely unseen.
For a long time, many tiny things around us remained unknown. People discovered that a curved piece of glass could make small things look bigger. The invention of
Look at the letters through the water flask. Do you notice that they appear larger? It acts just like a magnifying glass. Now use a real magnifying glass to look at an ant. Can you see its body details more clearly?
In 1665, Robert Hooke published Micrographia. He was a careful observer. He showed detailed drawings of tiny things people had never seen before. His microscope made things look 200 to 300 times bigger than what one could see with the unaided eye. One day, he looked at a thin slice of cork and saw it was made of many small, empty spaces. These compartments reminded him of a honeycomb. He drew what he saw. Around the same time in the 1660s, Antony van Leeuwenhoek, a Dutch scientist, made better lenses that allowed him to build more useful microscopes. Around the same time in the 1660s, Dutch scientist Antony van Leeuwenhoek he was the first person to clearly see and describe tiny living things like bacteria, blood cells. Because of this, he is known as the father of microbiology. All living beings are made up of cells. You might wonder what cells actually look like. Let us take a closer look at the basic structure of a cell using a microscope.
First, grab a fresh onion bulb from your garden or kitchen. Now, turn on the tap and wash it thoroughly with water. Make sure to scrub away all that excess dirt. First, we take a fleshy piece of an onion. Watch as we gently pull away the thin epidermal layer. This transparent layer is called the onion peel. Place the peel in a petri dish containing a few drops of safranin for 30 seconds. This will give the cells a pinkish color and help us see them clearly. Now, we place a drop of glycerin over the onion peel. This will prevent the cells from drying out. It also improves clarity for better visualization under the microscope. Place the cover slip at a 45-degree angle. Use the needle to lower it slowly to avoid air bubbles. Gently withdraw the needle as it settles flat.
Use blotting paper to gently wipe off any extra glycerin. Do this carefully around the edges of the cover slip. You will observe nearly rectangular structures under the microscope. These are plant cells, with clearly visible walls and chloroplasts.
These are the cells of the onion peel, which are closely arranged without any space between them. Let's observe the leaf peels of different plants. Carefully peel the epidermal layer from the leaf. Mount it on a slide with a drop of water. Now, let's examine it under the microscope.
you will find that all plants are made up of cells. What do you think the body of an animal is made of? It's a vast world of cells, billions of tiny building blocks, working in perfect harmony.
Hey everyone! I hope you enjoyed this content. Please like and share this video, and also subscribe to my channel and blog.